All right, so welcome back. I hope everybody had a nice break. I just got back from a 9-hour road trip yesterday, so that was my fault. That was a bad call, but I hope everybody had a nice restful break. But a couple of things before we get started. I did send out an email with your grades. Hopefully you saw all that and I didn't ruin your Christmas too much. But there was a bit of a curve on the exam number 1. There were 2 true-false questions that almost everybody got wrong. So that indicates to me that that was a problem with either the question or the way that I taught it, if everybody gets it wrong. So those 2 questions were curved, which added 3 points to the exam score, which should have been already reflected in your Canvas grade. A reminder, just a general reminder, that I do have an option at the end of this class. The last day of class is an optional final. So if you didn't do as well on this exam number 1 as you would have hoped, there is always the option to take that comprehensive or cumulative final, and that final replaces your lowest test grade. So not everything is lost if you didn't do as well as you wanted to on this first exam. People usually find the first exam to be pretty difficult. I think it's just adjusting to a new class and note-taking style and test style. I like to think that my tests are pretty straightforward, but sometimes it takes a bit to get used to a particular professor's teaching style. So again, not all is lost. If you want to take the comprehensive final on the last day, you are more than welcome to. And that comprehensive final can never hurt your grade. It will only help it. So if you do worse on that test than you did on your other tests, it's just like it never happened. Okay, second thing, I don't hand back my tests in class. If you would like to look at your test, you're more than welcome to come by my office. You can look at your test and you can ask me questions about it, or you can just quietly look at it, whatever way you want to do it. But yeah, I don't hand them back. I do recommend that everybody come look at their tests, especially if you didn't do as well as you expected. Like if you left that test feeling like you got a solid B and you got it back and it was a D, you need to figure out why. Where was that disconnect? And you're only gonna get that information by looking at your test. And yeah, so you're welcome to schedule an appointment or just come by during my office hours, but I will not be handing back any of your exams. All right, and then the last one is I don't know if it's something we've talked about, but it's just a reminder, something to think about, is that so I do attendance by code, right, by QR code. There's also just the link there. Sending the links to somebody who's not in this classroom is academic misconduct. Now, is it a huge breach of academic misconduct? No, but when you get reported to the Honor Council, they don't care. So sending this code and letting somebody who is not sitting in this classroom say that they are sitting in this classroom is academic misconduct. And I have a sneaking suspicion, judging by the fact that there are about 80 responses right now, and I don't think that there are 80 of you in here, that there are people that are doing this. Now, I can't prove it right now, but at some point in the next couple of days, I will be sending around a sign-in sheet in addition to this. And so if I have somebody who did the QR code and didn't sign the sign-in sheet, that's indicative that they are not in this room, okay? And I will be reporting you to the Honor Council if you do that. So remember that points in this class they're worth something, but they're not really worth something. If you don't want to come to class, don't come to class. It's not going to hurt your grade. Don't fake it, okay? That's the big problem. Not coming to class is not going to get you in trouble. What will get you in trouble is faking it. Okay, so if you're I know probably if anybody's faking it, it's not you guys sitting here. But for that to happen, somebody else has to text the code to somebody. Okay, so if you are texting codes to people, stop. It's not worth it, especially for something that is not going to affect your grade. It's not worth it. Okay, and I will be doing spot checks in the next couple of days and then probably a few more times before the end of the quarter. And it will be random. You don't know what part of class it's going to happen in. Or which day it's going to happen. All right, so that is my warning to all of you before I actually check it, okay, that I do consider that academic misconduct. You are lying. Whether it's a big lie or a small lie, you're lying, and it's academic misconduct. Okay? Everybody cool with all that? That all makes sense? Okay. All right, so we are going to pick up, does anybody have any questions about anything that I just talked about? About the exam, the final exam, anything? All right, so we are going to pick up right where we left off. We left off last time before the break, and I know that was a while ago, but we left off last time before the break in chapter 6, and we're talking about microbial nutrition, which is just how microbes eat, how they grow, how they divide, how they reproduce. All right, last time, we talked about all of these environmental factors that go into how microbes grow and how microbes are affected by different environmental procedures and different environmental conditions. And so we talked about, there are 3 or 4 major ones. We talked about temperature last time. We talked about the ranges at which they grow and the names associated with each of those. And then I wanted to see. Okay, looks like most of you got the correct answer on my poll. It was most human pathogens are in what category of optimum temperature, and the correct response is mesophile, because mesophiles are right about human body temperatures. So bacteria that like to grow inside human bodies will do best at human body temperature. Okay. Now we are going to move on to our next one, which is pH. So what pH is, if you don't know, is pH is the measure of how acidic or alkaline, and another word for alkaline, I don't know why everything has to have multiple names, but another name for alkaline is basic. So how acidic or basic something is. And there's a lot more complicated reason for this, like why something is acidic or why something is alkaline, has to do with hydrogen ions and all kinds of stuff that we're not going to talk about. All you have to remember for now is that pH is a measure of how acidic or basic something is. And the pH scale goes from 0 to 14. It's a flat scale from 0 to 14. And 0 is the most acidic you can get. So the lower the pH, the more acidic it is. And 14 is the most basic or alkaline something can be. Okay? And then 7, which is right in the middle, is what we consider neutral. So it is not acidic or basic, it's neutral. Right? So 7 is neutral, 0 is acidic, 14 is basic. So, and this should make sense, neutral, our most commonly associated neutral substance is always going to be water. Water is considered perfectly neutral. Okay, it is not acidic or basic, it is neutral. Some examples of things that are acidic are things like gastric acid. Gastric acid is the acid inside of your stomach. It has a pH of very close to 1. It's a little bit less than 1, so it's extremely, extremely, extremely acidic. And then lemon juice, orange juice, tomato juice, a lot of fruit juices are acidic, citrus things are acidic, vinegar is acidic, things like that. And then alkaline or basic are things like bleach. Bleach is extremely alkaline. Ammonia, baking soda, even seawater is slightly alkaline. Now, human blood, which is not it's not listed on here, human blood sits right about there. And the pH of human blood is kept in a very, very, very tight window of 7.35 to 7.45. Okay, your human blood is constantly kept in that very, very, very narrow pH range. Okay, your body would cease to function if it fell out of that pH range. Okay, that's why you have kidneys. Your kidneys spend all of their time keeping your blood in exactly that pH range. Bad things happen if it gets too acidic or too alkaline. Okay, so that's why things like, one of the things that just drives me crazy, and if you like the taste of it, you like the taste of it, that's totally fine. You're allowed to like the taste of it. But things like alkaline water are complete garbage, right? They don't change anything about your body chemistry because you have working kidneys. okay? And if your body was alkaline, you would be extremely, extremely, extremely ill, okay? So alkaline water is not going to do anything. If you like the way it tastes, that's great. You can drink it. But you can do whatever you want. I'm just trying to tell you that it's not actually going to do anything clinically to your body. So your human body is right around neutral. It's slightly basic, but we usually consider that neutral because it's not even to an 8 yet. All right, so in terms of bacteria, there are going to be bacteria that can grow at all different pHs, right? Different bacteria are going to like different pHs. If an organism likes acidic pHs, we call it an acidophile, which should make sense. Phile means liking or loving. Acidophile means it likes acidic environments. There are certain bacteria that like acidic environments, particularly something like lactobacillus. Now, does anybody know where lactobacillus lives in the human body? Nobody want to say it out loud? It lives in the human vaginal tract. Lactobacillus lives in the human vaginal tract. The human vaginal tract is slightly acidic. So the human vaginal tract usually sits around a pH of about 4. And we'll talk about why that's necessary when we get to the human immune system. But just know for now that vaginas are typically kept at a pH of about 4, which means that the bacteria that lives in them, like lactobacillus, again, I'm not writing it down so you don't have to remember that name, like to grow at a slightly acidic environment. They're acidophiles. On the other hand, you have alkalophiles, and alkalophiles like alkaline environments. Okay? A really good example of this is something called Vibrio. Now, Vibrio lives in seawater, which is usually between 8 and 9 pH, depending on how salty it is. But it lives in a pH of about 8 to 9, which is alkaline. So they are alkalophiles. And there are bacteria that can live in even more alkaline environments. But if you think about it, most bacteria, do you know of any bacteria that can survive living in bleach? Nah, bleach kills just about everything. So there's not usually bacteria that go all the way to the complete spectrum of alkaline, but there are some that like to live in a slightly alkaline environment, and we call them alkalophiles. Now, based on what we talked about with temperature, which category do you think most human pathogens fall into? The only one we haven't talked about yet. Neutrophiles, right? So the last one are neutrophiles, which are bacteria that tend to live at neutral pHs. Now again, most of the inside of the human body, most of your organs and blood are kept at a neutral pH. So most human pathogens are going to be neutrophiles because they grow at the neutral pH of the inside of a human body. Now again, there's certain exceptions to that. Again, like lactobacillus grows in an acidic environment. There are some bacteria that can grow inside of your stomach. The inside of your gastrointestinal tract, like we talked about, has a very extremely low pH. But most of the time, our human pathogens are going to fall into this neutrophile category. Right? So that is pH. We have 3 categories. Acidophiles like to grow in acidic environments. Alkalophiles like to grow in alkaline environments. And neutrophiles like to grow in neutral environments. All right, and the last one, I think, that we have is oxygen requirements. So this is going to be the last big category of how organisms grow in particular environments. So oxygen requirements, again, the kind of fancy word for this is the gaseous composition of the environment. And there are going to be 5 categories of this organism. Before we can talk about the categories, I do want to talk about this kind of special media that we have here. So these tubes, these pictures of tubes, and I'm going to talk about all their growth patterns as we go through, but these tubes are something called thioglycolate broths. And thioglycolate broths are specially designed to have what we call an oxygen gradient. What that means is that they have high oxygen at the very top, which should make sense, right? Because the top of that tube is exposed to the environment, right? And you have lots of oxygen in the natural air. Okay? In our natural air, we have tons and tons and tons of oxygen. So that very top of the tube has basically the same amount of oxygen as we do in the environment. Okay? But then as you go farther and farther and farther away from the top, there's going to be less and less and less oxygen. And by the time you get all the way to the bottom of the tube, there is no oxygen. Okay? So the very top of this tube has atmospheric amounts of oxygen, tons and tons and tons of oxygen, and the very bottom has none, if these are properly prepared. There's no oxygen at the bottom. And no oxygen is what we call anaerobic. Okay, remember when you put an A or an an in front of a word, it means without. So anaerobic means without oxygen. Okay, so the top of the tubes are aerobic, meaning they have oxygen. The bottoms are anaerobic. Okay? So to demonstrate this, we're going to look at where these bacteria would grow within this tube based on their oxygen requirements. Okay? So our first category is going to be obligate aerobes. Okay? And again, look at the word. Obligate means it must. It has to. It has no choice. It has an obligation. Obligate aerobe means that it has to have oxygen. It has to have lots of oxygen. Aerobe or aerobic means oxygen or oxygenated. So obligate aerobe means lots of oxygen. It must have lots of oxygen. These types of bacteria are not going to grow in any place that has less than atmospheric oxygen. They will only grow when there is the same amount of oxygen that we have in our atmosphere. Okay? And so where that will grow in our little tube is it will only grow at the very top, which should make sense because that's the only place in this tube where there is atmospheric amounts of oxygen. The rest of this tube, there's not enough oxygen in it, so the bacteria won't grow there. Obligate aerobes. Now, on just the opposite end of that spectrum are obligate anaerobes. And these are bacteria who cannot survive in oxygen. Oxygen is actually toxic to them. So oxygen metabolism produces something called free radicals, if you've ever heard that term before. And free radicals are dangerous. And they destroy DNA. They can wreak a lot of havoc in any type of organism. Any organism that breathes oxygen or consumes oxygen has a way of dealing with free radicals, humans included. Our blood has tons and tons of enzymes and other things to get rid of those free radicals. Obligate anaerobes don't have those. So if they are exposed to oxygen, they will die. They cannot survive in any kind of oxygen. So the only place that these obligate anaerobes are going to grow is the very, very bottom of these tubes where there's no oxygen. Any other part of that tube is going to have some amount of oxygen, and they will not be able to grow there. They will actively die. Okay. Now, our next one is where we kind of get into the little bit of more complicated categories. We have a facultative anaerobe. And what a facultative anaerobe is they prefer to live in oxygen. And the reason for that, I can't remember if we talked about it, is that using oxygen for metabolism is more efficient. You produce more energy when you use oxygen. Okay? And a lot of these organisms can produce energy without oxygen, but it's less efficient. So they're going to prefer the one that is more efficient. They will prefer oxygen. So they prefer oxygen, but they can survive without it. So they can survive, they prefer oxygen. If they're given a choice, they will grow in an oxygenated environment. But if they are not given a choice and there's no oxygen, they will grow without it or with less oxygen. Okay, so the way that these are going to grow is there's going to be a big chunk of bacteria up at the top where the oxygen is, because that's where they want to be. That's their choice, that's their preference, is to grow at the top. But the little bacteria that are kind of pushed out from the top, when the top fills up and there's no more room, the bacteria will start to streak down, and there will be little bits of growth throughout the rest of the tube. Okay, but there will be a cluster or a clump at the very top because that's their preference. They have a preference. Okay. Now, an aerotolerant anaerobe is slightly different, but in an important way. It doesn't use oxygen, but is not harmed by it. Okay, so it doesn't use oxygen. It basically doesn't care. It's the bacteria, it doesn't care. It doesn't matter if there's oxygen, doesn't matter if there's not oxygen. It's not going to use the oxygen anyway, but the oxygen doesn't harm it. So the way that these will grow is they will grow equally from the top to the bottom of the tube because they have no preference, right? They can grow anywhere because they don't care about the oxygen. They have no preference either way. So they will grow equally from the top to the bottom. And then our last one are the microaerophiles. And these are kind of what we call Goldilocks bacteria. And the reason we call them that is because they can't have too much oxygen. They can't have too little. It has to be just right. Okay, like little Goldilocks. But it doesn't like atmospheric oxygen. Atmospheric oxygen is too much. But anaerobic is too little. So it has to be just the right amount. Now, the way that microaerophiles grow is they grow just under the surface of these thioglycolate tubes, right? So the obligate aerobes are going to grow at the very, very top because that's where the atmospheric oxygen is. But the microaerophiles grow just underneath the surface because they have to be our picky little babies, right? They have to have just the right amount. And those are the microaerophiles. And like I said before, I know that all of this seems really random right now, but in the next couple of chapters, I think it's the next section, we're going to talk about how we as humans capitalize on these environmental requirements to prevent bacteria from growing or to make them grow, right? Like we talked about over here, we take advantage of the fact that most bacteria can't grow in bleach, and we use it to clean stuff. That's how we prevent bacterial infections, right? Somebody in your house has norovirus, you gotta bleach everything in that place, okay? Because you're trying to kill the viruses. It's the same thing your body does. The gastric acid, there are very, very few bacteria that can survive in an acidic environment like your stomach. That is why all of the food that you eat goes immediately into an acidic soup to kill any bacteria that might be in there. Okay, and we're gonna talk about how we take advantage of pH, gaseous composition, and temperature to control microbial growth in every aspect of our lives. Okay, another thing with pH, most soapy water is alkaline as well. That's how soap kills bacteria or gets rid of it. All right, so we're going to be talking about this stuff a lot in terms of how we control bacterial growth. All right. So now that we've kind of talked about the requirements of bacterial growth, we're just going to talk a little bit about a couple of random ways that they do grow, kind of a couple of little loose ends that we have to cover. All right, so one of the ways that bacteria will grow, and it's an important thing to keep in mind as we're talking about bacteria, is most bacteria do not grow, or viruses, do not grow in an isolated way, okay? You're never gonna have just one bacteria hanging out on a surface, right? It's going to be, just like humans, we don't really live isolated. Aren't there a bunch of studies done that if you like isolate a human completely and they don't talk to somebody, they'll go crazy in like 3 weeks, right? We're not meant to be isolated beings, okay? Neither are bacteria. They grow in communities, just like we do. And so one of the ways that they grow, or the way that we categorize how bacteria grow with each other, is something called symbiosis. Now, symbiosis, as a definition, is a long-term association between two organisms of different species. Now, something that can be kind of confusing when we first talk about this is just from, like, in basic language, if I said the term, they had a symbiotic relationship, that automatically, to you, probably means something positive, right? When we say, oh yeah, that's really symbiotic, right? To us, we have kind of capitalized that word to mean something positive. But a symbiotic relationship is just a relationship. It's not positive, it's not negative, it's not neutral, it's just a relationship. So when I say that a bacteria and something else has a symbiotic relationship, that doesn't necessarily mean it's a good thing. Could be a bad thing, could be a good thing, could be a neutral thing. But symbiotic just means that there is a relationship that exists. Okay? So again, it's between two organisms of different species. So you can't really form a symbiotic relationship with a member of your own species. These are relationships between different species. And different bacteria and algae and bacteria and animals and bacteria and viruses and all of these different things form symbiotic relationships. The biggest symbiotic relationship we're going to talk about, and we're going to talk about it a million times, is the symbiotic relationship between a human being and the bacteria that live in and on them, right? There's all kinds of crazy stuff, a lot of really cool research being done on like gut bacteria and how they think gut bacteria is now tied to autism and Alzheimer's and all kinds of different crazy stuff. And so the relationship between the bacteria that inhabits your body and you and your health is a very important one. We're going to talk about it a lot. Now, within the category of symbiotic relationship, right? Symbiotic relationship just means any relationship. Now, we can categorize that relationship in a couple of different ways. There are actually more ways than just these three, but we're only going to talk about these three. Right? And the first way is through mutualism. So you can have a mutualistic symbiotic relationship, which is a positive relationship. It is where both partners in the relationship benefit. The example that's given here is nitrogen-fixing bacteria and leguminous plants, which just means bean plants. Okay, there are bacteria that live in the roots of bean plants. And the bacteria in that situation get food from the legumes. And the plant gets nitrogen, which is required for them to grow. The bacteria produce nitrogen, which goes into the plant, which produces beans, which the bacteria then eat. Okay, so it's a symbiotic, mutualistic relationship. They both have a positive view of this relationship. A good example for bacteria that is related to human health is the E. coli in the human gut. Okay, E. coli is a bacteria that lives inside of the human gut. It does a lot of different things for us. I think I've talked about this before, but E. coli in the human gut, one of the most important things it does is it produces vitamin K. Okay, vitamin K is an essential vitamin that is responsible for blood clotting, right? If you don't have enough vitamin K, you will bleed out of every orifice you have. Vitamin K is important to keeping your blood where it's supposed to be. E. coli produces a lot of that vitamin K for us. We do get some amount of it from diet and things like that. Leafy greens have lots of vitamin K, but a huge amount of the vitamin K in our human bodies is produced by E. coli. And then, so we benefit from that, right? And the way the E. coli benefits from that is they have a nice, stable environment with a steady stream of food, right? We eat constantly. So they live inside of our digestive tract, and every time we eat, they eat, right? And it's a nice, stable environment. There's not a lot of predators, or what would be a predator for an E. coli. So they live a happy life with a steady stream of food, and we get to not bleed to death, right? So we have a mutualistic relationship. We help each other out in ways that benefits both of us. The second type of association or symbiotic relationship that you can have is parasitism. And parasitism is exactly the opposite. It's when one partner benefits and one partner is harmed. So one is taking advantage of the other. And there are a lot of parasitic relationships that exist. Any type of human pathogen, the last time you had the flu, or a cold, or a stomach bug, or food poisoning, any of those things, you are being harmed, right? But the bacteria or virus that was taking over your body was having a grand old time. So they benefited and you were harmed. That's a parasitic relationship. Another one, this is a good example, is a human and a protozoa. So protozoas, we'll talk a lot about our eukaryotic organisms like Giardia, or worms, or anything else that lives inside the human body. And in the process of living inside of us, right, it's not like the E. coli where we all just live happily ever after and everybody gets what they want. When you have a tapeworm or a protozoa infection, the tapeworm is consuming all of your nutrients. People who have long-term tapeworms will be really, 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 really skinny no matter how much they eat, because the worms inside their body are consuming all their food. Okay, so the worms inside their body consume all of their food. They take all of their nutrients. You can die from a tapeworm infection from malnutrition, no matter how much you eat. But the tapeworm living inside of you is having a great time. It can grow to be 40 feet long and live there until it dies. Okay, so one partner is benefited and the other partner is harmed. And then the last one is commensalism. And commensalism is when one partner benefits, but the other is unaffected. Okay, and this one's kind of weird. It's kind of hard to come up with examples of this. But one example that they give is a bird and a tree. Okay, so the bird benefits here because the bird has a nice place to live, but the tree doesn't care. Doesn't matter. This happens with a lot of like little fish that sucker on to bigger fish. A lot of times the ones that sucker on, they don't hurt anything. They're just catching a ride, but they benefit. So one partner benefits, but the other one doesn't care. I'm sure that there are some examples of bacteria that live inside of our bodies that just don't affect us, right? They get the benefit of living inside a stable environment, but we don't care that they're there. They don't harm us. Okay. So those are the three that we're going to talk about. There are a couple of other ones that are even more confusing than these three, but those are the only three that you have to know. It is absolutely freezing in here. Wasn't it like 80 degrees in here before we left, and now it's absolutely frigid? Okay. All right, so one of the ways that bacteria can take advantage of symbiotic relationships is through something called a biofilm. And a biofilm is a synergistic ecosystem of microorganisms that work together to provide advantages over individuals. Okay, so there's a lot of mutualistic relationships happening here. And these bacteria, usually lots of different species, viruses, all of these things, they produce basically a colony or a village of different organisms that work together to provide advantages over individuals. Again, and think of this as like a human village. If you lived without any electricity or stores or whatever else, and you have 10 people in a village, are you going to live a better existence if every single person is gardening and raising children and baking bread and farming and all of this stuff? Or are you going to work out better, like every village has ever had in history, if one person's the baker, one person's the farmer, one person raises chickens, one person raises cows, right? You divvy up the work. That's what the bacteria do. They live in little colonies where one provides a certain type of food, one will remove waste, one will be responsible for providing the exoskeleton or the lipids and the proteins. Everybody has a different job. And they form these large colonies which work better, right? We work better as a team than we do individually. And biofilms can be very difficult to kill. Because they form these biofilms, right? It makes it difficult to get rid of them. A lot of times the only way that you can get rid of a biofilm is to physically remove it. Again, a really great example of this is the bacteria that grow in your mouth. You have to perform manual removal of biofilms twice a day, every day of your life, right? You brush your teeth every morning and every night, or you should. Brush your teeth multiple times per day. That fuzzy feeling when you wake up in the morning is the bacteria that have formed a biofilm overnight. And if you didn't physically scrape it off every morning, it would get bigger and bigger and bigger and more stable, and it would eventually rot your teeth out of your head, right? Those bacteria would consume all of your teeth enamel. It would drill holes through your teeth because they're working together to take care of that. And a good example of why physical removal is the only thing that happens is, have you ever had, right, you're hungover, you're staying at somebody else's house or whatever else, you wake up and you can't brush your teeth, but you try to do mouthwash? Does that do anything? No. It makes your breath a little bit better, maybe for a few minutes, but all that crap is still on there, right? You still have fuzzy teeth. It's a temporary solution at best. And as the bacteria gets more and more established, so if you went days and days and days and days without brushing your teeth, the mouthwash would do even less. The only thing you can do in that situation is physically scrape it off. So another example that I kind of give of this as like a real world example of human health is, this is what happened to me when I was in a car accident, right? We talked about all that. I had an infection, blah, blah, blah. At first, when I had the infection, they were able to basically perform, it's called an incision and drainage, right? So I have this huge abscess. It's like 16 inches long. It goes all the way from my hip to my knee. And instead of cutting my whole leg open to clean it out, they basically poke holes to get the pus out, which sounds really gross, but they poked 7 holes down the side of my leg and put little drains in to help all the crap come out, okay? That worked for a little while. I was feeling better. I was on antibiotics. And then about a month later, I got sent home. My infection started to come back. I had fevers again. I wasn't feeling good. And at that point, the only option was for my surgeon to cut open my leg. I have a wicked scar that goes from my hip to my knee because I had to cut it all the way open. And the reason they had to do that was, once they did that, my surgeon came to me afterwards and he's like, you had a massive biofilm on the inside of the tissue. And the only way to get it out, antibiotics weren't going to do it, drains weren't going to do it. We could have washed it. It's not going to do anything. The only way to get it out was to physically scrape it off. So when I was in surgery, he had to scrub the inside of my leg. I was knocked out for all of this, obviously. But he had to scrub the inside of my leg. He had to cut it open and scrub every nook and cranny to get that biofilm off. Because the antibiotics weren't going to work. Just rinsing it wasn't going to work. Because they did, like I said, I had holes in my leg. So they were like pouring stuff in one side and hoping that it was going to wash it out. But that didn't work. The only thing that was going to work was scraping it off. And there's a lot of situations where this happens. Things like people get biofilms on like heart pacemakers and things like that, and no amount of antibiotics is going to get rid of it. The only thing to do is to replace the pacemaker. Right? So we're going to talk about biofilms a lot when we talk about infections, and the ones that are capable of forming biofilms tend to be the most gnarly infections. So we're going to just kind of keep biofilms in mind. We're going to talk about them a lot as we go through the rest of this class. Right? Now I just want to talk a little bit about bacterial growth, how they physically look while they're growing, and how we as scientists describe and talk about their growth. Okay? And there are two major ways that we typically look at bacteria. We look at it in liquid media. And the way that we can tell bacteria is growing in liquid media is usually by cloudiness. Does anybody in lab remember the fancy word for cloudiness? Turbidity. Okay, turbidity is just how cloudy something is, basically. So this is an example up here. I can't remember if I showed you this picture or not, but this is a picture of human urine. So most human urine is clear, maybe slightly cloudy. If you have like bacterial cells, particularly if you have a vaginal tract, your urine tends to be a little bit cloudy. But for the most part, it should be clear. But when you have a bacterial infection, so you have bacteria growing in your urine can become what we call hazy, which is slightly cloudy, or it can become completely cloudy, which is what you see on the right. I've even had experience sometimes when people have really, really bad infections. Sorry, my hands are freezing. When people have really, really bad infections, the urine will actually become chunky, which is disgusting, but it becomes thick and viscous, almost like maple syrup. Because the bacteria gets thicker and thicker and thicker and cloudier and cloudier and cloudier until it's thick. Okay? And then another way that we look at bacteria is through solid media. And usually what you're looking at on solid media are colonies. Okay, so this is a picture of it doesn't matter what type of plate, but that's an agar plate. And those on the right, the left side, the little gray dots, and then on the right side, the little pink dots are bacteria. They're growing in colonies. Okay. All right, so bacteria, for the most part, replicate through what's called binary fission, okay? And you don't have to memorize all the steps. All you really have to know about binary fission is, again, look at the word binary, meaning two by two. Binary fission. Fission is the opposite of fusion. Fusion is when you fuse two things, right? You take two things and make it one. Fission is the opposite. So you take one thing, break it into two. So you have one bacterial cell, and it basically replicates its DNA and splits in half. That's it. You don't have to memorize all the steps or their names or anything like that. You just have to memorize that it goes from one cell to two. Okay, and that is one round of binary fission. Going from one bacteria to two is one round of binary fission. Now again, this is asexual reproduction. There is no exchanging of DNA happening here. It's not required for bacteria to replicate. So when bacteria replicate, you will have one copy that splits into two genetically identical copies. Now, when we talk about bacterial growth, we tend to talk about them in their doubling or generation time, okay? So it's very common to talk about animals and other species and things like that in terms of their generation time. And what generation time is, if you don't know, is it's the amount of time it takes to go from being born, basically, to birthing another human. So if you're looking at it in humans, it's the time it takes. Your generation time is the time it takes from you to be born. The minute you're born, your generation time starts, to the minute that you produce another organism. Okay? On average, a human is about 23 years. Now, we can produce humans at different times, right? People become sexually able to produce humans when they're, 13? But we like to hope that they don't do that. But generation time and doubling time is different for every species, okay? It has to do with gestational time and the age at which you reach sexual maturity and things like that. But bacteria reproduce much faster than animals, okay? Animals, it takes years and years and years. Even the fastest animal that's capable of reproducing, it takes them years to become sexually mature, or months at the very least, okay? But bacteria are capable of doubling in a much faster time period. The average time, so I guess we'll talk about fast ones. So E. coli, which is kind of an average bacteria, it is, So E. coli is kind of a typical bacteria. It consumes most of our typical things. It lives in typical environments, right? E. coli is kind of an average bacteria. Its doubling time is 20 minutes. Okay, so you can go from having one cell of bacteria to having two in 20 minutes. In 20 more minutes, you'll go to having 4, and then 8, and then 16, right? Because every single one is gonna double every 20 minutes. That is how, if you're in lab, you can go from this, a perfectly clear broth, to a completely cloudy broth in less than a day, right? Some bacteria do have different doubling times. There is some variation there. 20 minutes is kind of standard. E. coli is about 20 minutes. The longest one that I know of is a bacteria called M. leprae, which stands for Mycobacterium leprae. It's the causative agent of Hansen's disease, which used to be known as leprosy. And its doubling time is about 2 weeks. Okay, so when you compare 20 minutes to 2 weeks, that's a very different type of bacteria, right? Mycobacterium leprae takes years to establish infection. E. coli takes a couple of hours. Okay, that is partially why if you get E. coli food poisoning, it starts to show up, 6 hours after you eat it? Right? You eat bad food, it takes a couple hours before you start to throw up your guts. Right? It's because E. coli grows really, really fast. Okay, so that's what we're talking about when we're talking about doubling time. It's the amount of time it takes one bacteria to double itself, to go through one round of binary fission. Okay, now I want to look at the growth curve for most bacteria. Now, when we're looking at this growth curve is assuming a closed system. So this is mostly the case. What we're mostly talking about here is bacteria in a tube is usually what we're talking about. It's something closed and no food is coming in, no waste products are going out. It's the equivalent of you sitting in a closet with a certain amount of food, okay, and seeing how long you survive or what happens to you in there. It's a closed system. Now, we're going to talk about how infections happen, and there are going to be a lot of similarities between this growth curve and how infections happen. But the inside of a human body is not a closed system, so it's not quite the same. But it'll look very similar when we get to that in a couple of chapters. So again, this normal growth curve is what is observed when a bacteria is growing in a closed system, in a tube where there's no extra food being added and there's no bacteria or waste products being taken away. Okay. And there are 4 phases. So what we're looking at here on the bottom axis, we have the number of hours. So number of hours from inoculation. So right here is inoculation. And remember that that is the process of adding bacteria to a media. So we're adding bacteria to the system. That's the inoculation. So that's our first data point. And then what we're looking at here on the up and down axis is the number of living cells. It uses the word viable. Viable means living or capable of living. All right, so we're looking at the number of living cells versus the number of hours that have gone by. Okay, so when inoculation happens, you add a certain amount of bacteria. So notice that we're not at 0 here at inoculation. We're a little bit above 0 because we added a specific amount of bacteria. We added some amount of viable cells. Now the first phase is going to be the lag phase. And the lag phase is what happens when nothing happens. Okay, the lag phase, the amount of bacteria is not going to change. Okay, and what's happening in this first little bit of time is the bacteria is producing food, it's consuming food, it's metabolizing, it's getting ready to reproduce. It can't quite reproduce yet, it doesn't have enough food or nutrients or anything like that. So no, the number of bacteria is not going up. Okay, it's going to stay the same. And that is going to happen for the first couple of hours usually. Okay, there's not going to be any change in the amount of bacteria in those first few hours. The bacteria are getting bigger, they're maturing, they're producing food, things like that. Okay? So that's stage 1 is the lag phase. The second stage is the exponential growth phase, and this is the phase at which the bacteria is actively dividing. They have tons of food, they haven't run out of nutrients yet, and so they are able to consume as many nutrients as they want and divide as many times as they want. So if this is E. coli, it's going to be dividing every 20 minutes for hours and hours and hours and hours. Okay, that's the exponential growth phase. And the amount of bacteria that's going to be in that tube is going to grow exponentially. It's going to double every doubling time. So in the case of E. coli, every 20 minutes, the amount of bacteria will double. And this, on average, to reach like peak exponential growth phase, so usually somewhere in the middle of that exponential growth phase, is going to take between 14 and 18-ish hours. Okay. Some bacteria will reach that point a little earlier, some will reach it a little bit later. It just depends on their doubling time. But the amount of time that we kind of shoot for in the lab is about 14 to 18 hours. Sometimes a little more, sometimes a little less. Okay, and then usually right around the 18-ish, 20-hour mark, the bacteria will reach what's called stationary phase. Now, what's happening in stationary phase is the number of bacteria that are alive is not changing. But the reason for that is because there is equal parts fission, which is division, right? Equal parts reproduction and death. So in the stationary phase, the bacteria are starting to die. They are starting to get to the point where they don't have enough food anymore. The original bacteria are getting old, right? There's only so many times a bacteria can divide before it dies. It's just the way that bacterial cells work. And so they're running out of food. We're actually having to start having a buildup of waste products. Again, think about a single human given a pack of fruit roll-ups and stuck in a closet, right? There's going to be a time where nothing bad happens. Everything is fine. They're eating their food. They're drinking their water. Everything is good. But at some point, they're going to have to go to the bathroom, and there's going to be a buildup of waste products inside this little closet. And waste products are not healthy, right? They're waste products for a reason. And the same thing goes for bacteria. They're producing waste products. The bacteria are getting old. They're running out of food. The box of fruit roll-ups is almost gone. And eventually, they will start to die just as fast as they're reproducing. So some bacteria are still reproducing, but a lot of them are dying. So we have equal amounts. So the overall number of bacteria is not going to change, right? It's going to go straight across, not going to change. Okay. And then at some point, usually around the 30-hour mark, the bacteria will reach death phase. Okay? And death phase is when you have more death than you do division. And so the amount of bacteria will slowly decrease until most of the bacteria is dead. Again, remember, they're running out of food. We haven't added any nutrients, so we're running out of food. We have a buildup of waste products. The bacteria don't have enough nutrients to metabolize and to reproduce. So they will just start to die. And eventually you'll get to a point where there are a few cells that remain viable for a long period of time, but we're basically back down to low numbers. Okay, it's just going to decrease over time. And again, this all depends on the type of bacteria. Some bacteria where a couple of cells will remain viable for weeks, right? You can stick a culture of bacteria in the fridge for weeks or months, and a lot of times it'll still grow months later. But yeah, those are the 4 parts. You have lag phase, where the bacteria are kind of gearing up to start dividing. The exponential phase, where you have the most amount of growth. The bacteria are all actively dividing. Nobody is really dying yet. Stationary phase is when you have equal amounts of replication and death. And then the death phase is when you have more death than replication. Okay. Now, something that is kind of weird to think about, but if you are running tests on bacteria, okay, so if you are going to be in the lab and you are trying to identify a bacteria, or you want to see how much food it eats or whatever else, there is a specific time in this cycle that you want to be testing the bacteria. And it is in the exponential growth phase. And the reason for that is, think about yourself. If I was going to give you a test, are you going to do better on a test if you are well fed and well rested, you had enough space, nobody's bugging you, your nervous system is calm, right? You had a nice breakfast? Or are you going to do better on a test if you had 3 gallons of coffee and you're jittery as hell, you didn't sleep all night, your roommate's bugging you, somebody left their dishes in the sink, right? If your immune system or your nervous system or whatever else are all gone to crap, you're going to feel like garbage and you're not going to do as well on a test, right? You're going to do better on a test, you're well rested, you feel comfortable and calm. It's the same thing with bacteria. They do better on tests, and I know it's not the exact same thing, but they act the best when they're the happiest, right? And the bacteria are the happiest in that exponential phase. It's where they have plenty of food, they got plenty of space, they're reproducing happily. Okay, so that is why when we go into lab, that's why I have to come in every Sunday and make bacteria for you. So if you're in lab and you're in lab on Monday, those bacterial cultures were plated on Sunday. I can't plate them on Friday because that is too many hours. If I plated them on Friday, by the time we got to Monday, we'd be past their happy stage, we'd be well into their death phase, and they're not going to act the way that we want them to. Okay, so when you're running any kind of tests on samples in a hospital or in a research lab or anything else, bacteria need to be in that happy exponential phase. And like I said, most bacteria reach it approximately 14 to 18 hours. So most cultures, when you're using them in a lab, we want them to be about 14 to 18 hours old. Sometimes a little more, sometimes a little less. But if they get too old, they're not going to act right. So that's something to keep in mind when you're in lab. Okay. Now, there is a lot of reasons that we would want to analyze how many bacteria are in a sample. Okay, so analyzing population size is just a fancy way of saying how many bacteria are there. Okay, because bacterial infections that are happening inside of a person, or bacterial that are growing in any type of environment, and I'll give you some examples in a second, But this is not the kind of thing where, that there's like that joke that you can't just be a little bit pregnant, right? You can have a little bit of an infection. There is a difference between having a few bacteria and having millions, right? You can have a little bit of an infection. You can have a medium bit of an infection. You can have a really severe infection. There are layers to this, and there is a spectrum. So when we are trying to diagnose somebody with a bacterial infection, we have to know where on the spectrum they exist. And so there are certain cases where we do need to quantify or put a number to how many bacteria are present in a sample. Okay? Another reason that that's really important is for doing like environmental microbiology, right? So bacteria is everywhere. We've talked about that before, that bacteria is in and on everything in the world. And most industries exist by controlling bacteria to a certain point, right? Like there's allowed to be bacteria in your drinking water. It's not sterile, there is bacteria in it, but to be considered safe, it has to have lower than a certain amount, right? The same is true for makeup or medicine or anything else, shampoo and lotion. Those are all going to have a certain amount of bacteria that's considered okay. Food products, milk, juices, they all have bacteria in them. It's just a matter of how much, and how much bacteria is considered appropriate for what that is. Okay, so in those cases, again, people who run water treatment plants or who produce food or who produce makeup or who produce any other kinds of products, medicines or supplements or anything else, a big part of their production is measuring the amount of bacteria. And if the amount of bacteria gets too high, then they can't sell that product to you. Okay, that's what the FDA does, is they monitor how much bacteria and stuff there is in all of those things. So there are a lot of different reasons why you would want to figure out how much bacteria there is. And we're going to talk about a couple of different ways that we do that, how we count the number of bacteria. Because as we've established, bacteria are very, very tiny. You can't see them. It's not like you can just count them, right? And that would not, even if you could, counting the millions and billions and trillions of bacterial cells is not a normal thing to do, right? Not going to waste our time. That is not time efficient. So there are a couple of ways. We're just going to talk about a few. There are a lot more ways than the ones we're talking about here, but these are the ones we're going to talk about. So the first way, and the way that is most common, I would say, not the most common, but one of the more common ways that's used in medical labs, this is pretty common in medical labs, is measuring turbidity, right? Which is the relative cloudiness of a sample, how cloudy something is. So if we go back to this one that we had a couple of slides ago, this person who has clear urine, they don't have an infection, probably, because there's almost no bacteria in there. This person who has hazy urine has a medium infection, if we're going to put that word to it. And then this person who has really cloudy urine has a very severe infection. The person with the cloudier urine has more bacteria in their urine. Their infection is more severe. And so I can look at this and say, oh yeah, the cloudy one is more, if we're putting numbers to it, like this is 1, 2, and 3. 3 is more cloudy than 2. 2 is more cloudy than 1. I can say that they have a relative, 3 relatively has the most severe infection. Number 2 has a medium, and number 1 doesn't have an infection at all. But that is just what I can do with my own eyeballs. If things were really close in cloudiness, I wouldn't be able to tell because my eyes are not designed to do that. So what we can do is use something called a spectrophotometer. And a spectrophotometer basically just measures the cloudiness of a sample. That's all it does. It's not going to tell you, the thing about this is it is not going to give exact numbers. What we're not going to be able to say here is, yeah, there are 500,000 bacteria per mil in this sample. All we're going to be able to say here is this one is more cloudy than this one. Okay, so it's a relative, qualitative measurement. So a spectrophotometer basically just uses light. It shines light through the sample and measures how much of that light gets blocked. Okay, if something is more cloudy, it blocks more light. If something is completely see-through, like this right here, most of that light is going to go straight through the tube because it's see-through, right? Think about looking through a window, right? A window that is completely clear is going to have almost all of the light come through it because it's completely clear. But something that has a lot of cloudiness or turbidity, that light is going to bounce off. And only very little of it is going to go through. And that's how a spectrophotometer works. It's like the difference of looking through a window, like a clear window, and looking through a frosted window. A frosted window is going to allow you to see some things, but only very little light is going to come through. And then looking at a door. A door is so cloudy, turbid, that you can't see through it at all. It would block all the light. Okay, so a spectrophotometer is just a way of actually measuring the amount of cloudiness rather than relying on my own eyes to do that. Okay, and then these three things here, or I guess these two, we'll say the first two, 1 and 2. These are going to actually count the physical number of cells. Okay, so these are what we call quantitative, which means that they are going to give actual numbers. Okay, and there's two different ways that we can do this. The first one is through direct cell counts, which is actually manually counting the number of bacteria. You take a small amount of any sample that you want to look at, and you put it in this special slide. We're not going to talk about what that slide is called, but you put it on a special slide that has this funny looking grid on it. And then when you look through the microscope, you use the grid to physically count the number of cells. And you basically multiply that out. It's determined that the amount of liquid on this slide is like 0.001 microliters. And so if you count 10 bacteria on here, you multiply it out, doesn't matter. You have 10 million bacteria per microliter. But you will have a physical number of bacteria per amount of sample. Okay? This is really common in fluid samples in a medical lab. Because especially things where bacteria is not supposed to grow. So in things like the fluid around your heart, or the fluid in your spine, or the fluid in your joints, right? The fluid in your body is not really supposed to have bacteria in it. Your blood, your cerebrospinal fluid, the fluid around your organs, your abdominal fluid, even your urine is not really supposed to have bacteria in it. And so if it does, there's probably not going to be very much. And this is a really easy way to give an exact answer of how much bacteria is growing in this person's heart, right? That's an important number to know. We know on exact numbers here. Another one that is commonly used is something called a Coulter counter. And a Coulter counter, again, you don't have to know the exactness of how this works, but it's an automated way of counting liquid samples. And it works through counting charges, basically. But you put the bacteria in some kind of liquid, and it travels across this detector. And this detector is going to count the number of bacterial cells. Again, how it does it, you don't really have to know. All you have to know is it's an automatic counter. Okay? And so it'll physically count the number of cells as it goes through this tube. And this is very commonly used for water and product testing. So this is how they test water samples this is how they test, millions of gallons of water for the number of bacteria, right? Because when you're testing like a water sample, like is this water bottling plant producing water that doesn't have too much bacteria in it? You take a small sample of it and you just run it through this automatic counter, it counts it, and you know what numbers are okay, and you compare it and you're done. Rather than having to individually count each and every sample and all that fun stuff. And then the last one is one that is not very common as of right now because it's very, very expensive, but it is very effective and very accurate and relatively simple. And this type of counting of bacteria and viruses is going to become, in your guys' career, it is going to become much more common. So that's kind of why we talk about it here. We are just in the beginnings of being able to use genetic instruments to measure the amount of DNA. So that's what these do, is it uses instruments to analyze and quantify the amount of bacterial or viral DNA present. So if you measure the amount of bacterial DNA, if there's more bacterial DNA or less bacterial DNA, you can determine how much bacteria there is. Or viral DNA. And so this, I think we talked about this at the end of viral testing. But this is going to become very, very common as you guys move into your careers. It's very expensive right now. It's not commonly used a lot of times. There are a few examples, things like, and we'll talk about why this is true much later, but gonorrhea and chlamydia are sexually transmitted infections, and they're actually really hard to test for using traditional methods. So in the last 5 or 10 years, they've switched to using genetic probing for those. But as of right now, that's still really expensive. It's not entirely accessible. And so it's not as common as I think it will become in the next few years. But genetic probing, again, is just quantifying the amount of DNA present using special instruments. Okay. All right, that is where we will stop today.